Hello people, hi guys, good morning, good afternoon from anywhere you're watching this from. My name is Tolulope Oladimeji, and if you are a returning viewer on this channel, I am sure you can say that it's been a while you've seen my face on here, and this is largely due to some life changes. The last time I was here, I was rounding up my master's program from a university here in the United States. But right now, I work as a scientist in a biotech company, doing some of the most amazing work in biopharma. So largely due to this change, and also because I would like to reach a wider audience, my videos moving forward will focus on sharing career tips and also explaining and discussing about lab techniques that you can use to position yourself for an advanced career anywhere in the world and not just in the United States. Without further ado, today's video will focus on explaining a very, very crucial technique in molecular biology, which is called polymerase chain reaction, or PCR for short. From the name of the technique, we see that there are two major key terms in that technique name. There is polymerase and there is chain reaction. Polymerase, which is an enzyme. So quick one, let me give you a pro tip. In science, words that typically have the suffix "ase" refers to an enzyme. Words like nuclease, like helicase, like ligase. Even though not all enzymes have that suffix, but most words that have that suffix "ase" is referring to an enzyme. While chain reaction, because in each step of the reaction, the product is the template for another reaction to happen. So we know that when you start, for example, one DNA strand from the beginning of your PCR, you double that, and those new DNA strands then become the template for another reaction to happen. Hold it, I know that it might begin to look confusing, but trust me, PCR is not hard. At least in theory, it should be quite easy to comprehend, and that's what I hope to achieve with this video, help you comprehend it better. So the first thing you need to do to avoid confusion or to make this simpler is to understand that PCR is actually science way of mimicking what naturally happens in your system, in your cells. We know that during the process of cell division, mitosis or meiosis, what your system is trying to do is double the number of your cells. And when you double the number of your cells, what are the components of those cells? Your DNA and other organelles. So that process of being able to copy your DNA from the starting parent cell to daughter cells is actually what I'd like to think of like the natural PCR. And what we do in the lab is then the artificial PCR. You get it? Okay, so now that we know that PCR is not strange to your system, it's going to be very, very easy from on here to actually comprehend this really well. For your polymerase chain reaction to start, you need the deoxyribonucleic acid material from any biological source, maybe a plant, an animal, or even a microbe. Remember that the DNA is a double helix structure. But for the reaction to progress, we need to go from this double helix to two linear strands of the DNA. And how do we actually achieve this in vitro during PCR in the lab is that we apply a very high temperature to denature the non-covalent bond that is actually holding this double helix together. So the non-covalent bonds like hydrogen bond. It denatures that double helix to two linear DNA strands. I can imagine that as far back when PCR was first discovered by Karimulis, they actually went through a lot of steps trying to find the optimized temperature to do denaturation and every other steps following. In fact, what they did then was that they took the biological sample, the DNA, and moved it from one water bath of, let's say, 98 degrees Celsius to another water bath of a lower temperature to continue the steps. But in today's lab, you can actually complete this process in a very fancy machine called thermocycler. Thermocycler is just a fancy name for a machine that is able to regulate its temperature from as high as 103 to as low as 4 degrees Celsius. So things have become really simpler and easy, and they can only become more simple and easy from here on. In vivo, during DNA replication, which I call the natural PCR, how the DNA unwinds from the double helix structure to the linear structure is through an enzyme called helicase, because we obviously don't want our cells heating up to 98 degrees Celsius. So once we have a fully denatured DNA template, we need a material called a primer to mark out the region of our DNA that we hope to amplify with the polymerase chain reaction. A primer is a short sequence of DNA that match with a particular sequence on your template DNA. So you can think about a primer like a probe. It's supposed to go through your DNA sequence and find where it matches. Remember that your nitrogenous bases, the adenine matches with the thymine, the guanine matches with the cytosine in specific sequences. So your primer probes for your part of your DNA that matches with its own short sequence. It's about 18-20 bases long. Primer design is something that is very technical and something that you have to be very, very intentional about before you do your PCR, because it will determine the success or otherwise of your reaction. If you're working on a widely researched genome like bacteria or fungi, you can easily find sequences of primers that will work online, then send this sequence to primer manufacturing company like IDT, Thermo, or Inkaba, depending on the region where you are, and they'll have this delivered to you and you can then use it for your reaction. However, if you're working on a less popularly researched genome, you might actually have to design your own primer. Primer design is a very crucial topic on its own, and I would really love to elaborate on this. If you guys are interested, kindly let me know in the comment section. Something else we want to think about regarding primer is that some DNA samples that you have, you might not essentially know what genome it is representing. You don't know if it's a bacteria, if it's a plant, or it's a human genome. But you can use a primer to actually figure out what it is. So for example, there's a popular gene that bacteria have, which is called the cysteine gene. If you use a primer that would specifically align to this cysteine gene on your sample and it amplifies, then that tells you that your sample actually contained a bacterial genome or otherwise. So this step of your primer binding to your DNA template is called the annealing step, and it happens within the temperature of 50 to 60 degrees Celsius depending on your primer. So the temperature is very specific to your primer, and how efficiently your PCR will work would really depend on how you have optimized your primers for the annealing step. For your PCR to progress, there are other reagents that need to be present in your reaction, one of which is the big guy, polymerase. Polymerase is an enzyme and it comes in right next to the primer after the annealing stage, and what the polymerase does is that it serves as an anchor for new nucleotides to be added to the newly formed DNA strand. Remember that in the law of base pairing, you have adenine binding to thymine, you have cytosine binding to guanine, and the dNTPs, or the deoxynucleotide triphosphate, that you need for this reaction to progress is actually going to be present in your PCR master mix. Other reagents that are present in your PCR master mix should be a buffer to steady the pH of the reaction. You also have magnesium chloride that helps the reaction to progress. So this step of the nucleotides coming in to the newly formed DNA strand is called the extension step, and it happens at about 72 degrees Celsius. So if you started with one DNA template, at the end of one cycle, you should have 2 DNA templates. And what do you think would happen next? You guessed it. Another denaturation, annealing, and extension until the cycles that you set up for is complete or all your reagents is used up. But typically, in an actual experiment, you don't want to start with one DNA template. That is why the concentration of your DNA before you start your PCR is actually very important. If the template concentration is too low, then the reaction will not be able to proceed successfully. And if it is too high, equivalent to the reagents that is present, let's say you have a lot of DNA template and very limited primer and dNTPs, obviously then the reaction cannot progress as it is designed to progress. I know this is a lot of information, especially if this is your first time interacting with this technique, polymerase chain reaction. But I want you to take a deep breath and rewind through this video. I have included chapters that you can easily go from one section, from the denaturation to the annealing to the extension step, to easily rewind and relearn again if you need to revisit this. If you learned a thing or two in this video, kindly engage with this video by liking, subscribing, and sharing this video to your network of friends that are also open to advancing their knowledge of lab techniques for an advanced career anywhere in the world. Thank you for watching.